week of our third grading. Okay, next week we'll be moving forward to the fourth grading part. Okay, so I'll be sending videos. Okay, so I'll be sending videos para po sa ating lesson na hindi na-cover. Okay, so once again, let us have today, let us have first a review of what we have discussed last week. Okay, so anyone who would like to give us a review of what have you learned? Anyone? Wala na akong kausap. Ilang mga anak, anong natutunan niyo last week? Hello?
Hindi replication po ba tawag?
Okay, so about DNA. DNA. What is the acronym DNA stands for? DNA stands for what?
Deoxyribonucleic acid.
Okay, so we have deoxyribonucleic acid. Okay, so it's part of the so-called Central Dogma. Okay, so once again, everybody say it. Acid.
Deoxyribonucleic.
Okay, replication ng sa 10 Mars. Times are called catalyst. means
Papabilisin.
yes, nagpapabilis, no? Okay, so nagpapabilis ng reaction. We call those as catalysts. Okay, then you will encounter also those words that ends in O's or OSE. Pag may OSE, okay, ano mo? What's our interpretation? They are called as what? Pag mga O's yung dulo, they are called as
Catalyst.
okay, so they are called as sugars. Ang mga asukal yun na. Okay, so pag mga O's yung dulo. Okay, so can you still see the presentation mga anak? Okay, so yung mga O's yung dulo,
Yes po.
we have, yes, thank you. Okay, so we have here the ribose, deoxyribose, pentose, maltose, saccharose, maltose. Okay, so yun ay mga asukal, no? Okay, so ang dami, no? Okay, so may mga O's yung dulo. Okay, now sa DNA replication, you have already watched this one, right? Napanood nyo na, di ba, yung sa DNA replication? Tama ba, mga anak? Napanood nyo na to? Uy, kausapin nyo ako. Hindi nyo kinakausap. Okay, so once again, panoorin nyo yung replication, DNA replication.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, so that's DNA replication. No? Okay, so the DNA replication is being started by an enzyme called as ano yun? Anong enzyme yun? Anong enzyme yun, mga anak? Okay, so we call it as
Polymerase.
okay, so it's being started by the enzyme helicase, no? Okay, and it will create what? Anong sunod na mangyari? It will create a replication fork. Okay, then the replication fork has two, okay, so major strands. Okay, so what do you call the first one? Hello, mga anak. Hindi kayo nanonood. Okay, so you have watched this for 3 times, 2 times. Okay. So, it will create two strands. Ano yung two strands na yun?
Leading strand, lagging strand.
Okay, yes. We have the leading strand and the lagging strand. Okay, so now the leading strand runs in what direction? The leading strand runs in what direction?
Kaliwa.
Yes. Anong direction ang takbo niya? Okay.
Pa left, sir. Yung
Direction niya is 3
3
prime.
to 5 direction.
Yes. 3 prime to. Okay. 3 prime to. What's the direction? 3 prime to.
5.
Yes. Okay. 3 prime to 5 prime. So that's the direction. How about the lagging strand? What's the direction for the lagging strand?
5 prime to 3 prime direction.
Okay, yes. Pabaliktad na. Okay, so 5 prime to 3 prime. Now, ang symbol ng prime is yung kuwit dyan, apostrophe. Okay, so we call that as the prime. So, maghihiwalay ito, di ba? Okay, so it will be creating two strands, the leading strand and the lagging strand. Direction of the leading strand is 3 prime to 5 prime. Okay, so and for the lagging strand, okay, so that's 5 prime to 3 prime. Okay, so now what's the case for the leading strand? So they can continuously create pairs and pairs of bases. Okay, so continuously. Okay, pero sa lagging strand hindi po pwede. Okay, so it will just create what? By fragments, no? Anong tawag natin doon sa bawat fragments? Sa bawat fragment, anong tawag natin? This is the helicase. It's the enzyme that starts the unzipping. Okay,
Okazaki fragment.
Correct. Okay, so we call that as Okazaki fragment. Okay, so Okazaki fragment is a Japanese term. Okay, so for a fragment being created in the lagging strand. Now, you will study this later, okay, so as you move on to senior high and to college. Okay, so ano lang tayo ngayon, basic lang. Yung yung pinaka tawag dito, yun na yun. Assignment ko to, right? Or nagawa niyo na to?
Sir, yung
Yes. Nagawa niyo na ba to or hindi niyo nagawa?
wala naman pong ganyan.
Okay. So, could you please get your notebook? Okay. So, rearrange. Just right there, A, B, C, D. And try to rearrange which comes first. Okay. So, write your name and the date today. Okay. Name and date today. Okay. Then, pakilagay sa inyong electronic portfolio. Sa e-portfolio. So, this time, which comes first? Okay, so yung una ay the DNA. Okay, so the DNA double helix breaks or unzips down the middle between the base pairs. Okay, so the unzipping is administered by what enzyme? Hello mga anak, anong anong enzyme 'yun? Yung nagano sa unzipping? The enzyme is ulit-ulit. Okay, so we call it as helicase. Okay, so yun. Okay, the second one, the enzyme DNA polymerase moves along the exposed strands and adds complementary nucleotides to each nucleotide in its existing strand. And third one, a complementary strand is created for each of the two strands of the original double helix. Okay, then the two new identical DNA molecules have been produced. Okay, so that's for DNA replication. Question? Any question for replication? Hello mga anak. Any question?
Wala na, sir.
Okay. Okay, so let's move on now to the next step is transcription. Okay, so transcription is the mechanism by which a template strand of DNA is utilized by specific RNA polymerases to generate one of the four types. Okay, to generate one of the four different types of RNA. Okay, so transcribe niya lang. Okay, so yun. DNA is used as a template for creation of RNA using the RNA polymerase. So transcription, ibig sabihin from DNA, gagawin nating RNA. So single strand. Kokopyahin lang, we'll just be copying. But copying the complementary pair. Okay, so please watch this one for transcription.
Proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. The motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called Intron Splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger.
Okay, so that's for the transcription, no? Okay, so from DNA to messenger RNA. So, ganun lang kasimple yan. Tatapatan lang natin.
Proteins are contained in our DNA.
Okay, so RNA polymerase reads the nucleotides on a template strand from 3' to 5' and creates an RNA molecule in the 5' to 3' direction that looks like the coding strand. Okay, so later on we'll be doing this later. The new RNA molecule is formed by incorporating nucleotides that are complementary to the template strand. Okay, so tatapatan lang. Yung G, okay, so di ba mayroong replication, magre-replicate siya. Ang kapartner ng G ay coding strand. Ito yung mother strand. Okay, partner ni G ay CC. Okay, partner ni T ay
CA.
A. Okay, so
A.
C, G, tapos A, T, T, A, T, A, C, G. Okay, GC, GC. Ngayon, okay, sa transcription, ano lang ang gagawin niya? Okay, so, yung na-duplicate dito ni DNA, okay, so, gagawa ngayon ng kopya para sa RNA. So, ang tawag na doon, it will become now a messenger RNA. Now, kukunin mo yung template strand. Ito yung template. Coding, then we have here the template strand. Now, anong partner ni C?
Si G po.
Okay, si G. Yung nasa green na. Yung ginagawa natin yung transcription na. Okay, so ang direction niya is 5' to 3'. Okay, so kanina ang direction is 3' to 3' to 5'. Okay, now, partner ni A. Anong partner ni A para sa RNA?
CU.
Yes. Huwag kakalimutan na, pag RNA, ang partner ni A ay U. Ang palatandaan nyo dito is AW. AW. Ay, AW. Okay. Dito yung mga AW. Okay, so that's the RNA. Okay, so AU. Siyempre, yung apat lang naman sila eh. Kung ang partner ay AU sa RNA, yung isa CG, ganun pa rin. Okay, so GC, GC. Okay, so ito yung magiging mRNA strand. That's for transcription. Okay, so transcription begins at the 3' end of the gene in a region called the promoter. Okay, so transcription begins with the promoter. Okay, so promoter, ibig sabihin yung promotor. Okay, transcription begins at the 3' end of the gene in a region called the promoter. The promoter recruits the protein. Okay, so TATA protein, TATA protein, or DNA binding protein. Okay, TATA. Okay, so DNA binding protein which in turn recruits other proteins. Okay, so yun lang yun. Okay, so begins what? Begins at the 3' end of the gene in a region called the promoter. Okay, next, the RNA synthesis involves separation of the DNA strands and synthesis of an RNA molecule in A. Ay, yung RNA, anong direction niya? 5' to
3'.
3' direction by RNA polymerase using one of the DNA strands as a template. Okay, so yun yun. Complete transcription of an RNA molecule. Transcription begins at the promoter, proceeds through the coding region, and ends at the terminator. So we have promoter, we have the terminator, or we have the beginning, and we have here the end. Okay, so parang ganun siya, start or the end. Codon. Okay, so we have here the exons and the introns. Okay, so hanggang sa maging isang protina. So later on, we'll be doing that. Okay, now, after transcription, it will, translation follows. Pag sinabing translation, kung anong nagawang mga bases, ito-translate mo lang siya into amino acids. Then, yung series of amino acids, yun na yung isang protina. Now, ilang amino acids ang kailangan sa isang protina? Hello mga anak. Kinalimutan na. Okay, so sa isang protina, usually there are 20 proteins, no? Okay, so para makagawa ng isang protina. So, ilang amino acid para makagawa ng isang protina?
20.
Okay, yes. Kailangan ng 20 amino acid, no? That's the usual. Pero dito, hindi natin kukompletohin. Okay, so sample lang yun nandito, sample lang. Okay, so transcription, ito yun. Ito yung ano niya, okay, sa coding strand. Okay, so then ito yung kapartner niya. So the RNA, no? Okay, so kukunin natin to tatlo-tatlo by pair of 3. Okay, so ang tawag dito. Group of 3, sorry. Group of 3. AAG. Okay, so yung first codon, AAG or first base. Okay, first group of 3. Then UCA, then GUC. Ngayon, hahanapan mo ngayon ng ano, these are codons. Hahanapan mo ngayon ng mga pangalan doon sa genetic code na pinag-aralan natin. Okay, napag-aralan na ba natin the genetic code?
Yes, sir.
Kabisado nyo na. Okay, so later on, we'll be doing that. So, dapat may mga screenshot kayo mga Okay, so, ng the genetic code. Okay, so codon, we have AAG, that's lysine. Okay, then UCA, that's serine. Okay. Ta-translate lang. Kung ano yung na-transcribe dito, ta-translate mo lang. So, ito yung grupo ng amino acid. So, yung grupong amino acid, yun yung isang protina. Di ba? Could you imagine that? Ganyan pala yung nangyayari sa ating katawan. No? Tulog tayo, tapos gawa ng protina sa ating katawan. Okay, ang dami pala nangyayari sa katawan natin, hindi natin alam, no? Okay, so, science lang yung nag-ano yan. Now, for translation, please watch this one.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Okay, so that's the last step now, translation. Okay, so to translate lang niya yung na-code. Okay, so into amino acid. Okay, then from amino acid, okay, yung grupo ng amino acid, then it will become now the specific protein. Okay, now, so let's review with the genetic code. Okay, so the genetic code, we have here the codon. Magkalimutan na yung triplet. Okay, so we have here the codon. Okay, so take screenshots mga anak. Mayroon na ba screenshots kayo nito? Okay, so
Meron na po, sir.
mayroon na. Okay, so ang ang pagpangalan, first letter nandito sa left, second letter nasa taas, third letter nasa right. Kaya pag sinabing A, U, tapos G. Okay, 3 bases. Okay, so A, U, G. Tatapatan mo lang. A nandito sa A, then sa U nandito, then si G. Kukonek mo lang. So therefore, okay, anong yun? Anong amino acid yun? AUG is
Met.
okay, so met. Ano ibig sabihin ng met? So dapat may screenshot kayo ng pangalan ng mga
Methionine.
Okay, yes, that's methionine ha. Okay, so that's methionine. Another, okay, GCU. Anong yun? Anong G tapos second letter C, U. Anong amino acid yun?
Alanine.
Okay, yes, alanine. Okay, so yung mga pagbasa ng dulo, mga nin lang yun. Alanine, methionine. Okay, so serine, leucine. Okay, so yun ang mga pagbasa na. So we have G, okay, another. UUG. Okay. UUG, ano kaya yun? Ano kaya ang amino acid yun?
Leucine.
Leucine.
UUG, yes, leucine. Okay, so leucine. No? Leucine or leucine? Okay lang yan. Okay, so far okay na, no? So yung tatlong bases na yan, pair of 3 or nucleotides in groups of 3, ang tawag doon ay codons ha. Okay, so then yung codons na yan, yun yung nag-specify ng amino acid. Diba? Ang galing ng mga scientists, no? Okay, so sa genetic code na to, meron tayong start codon. Ano yung ating start codon? We have the AUG, no? Okay, start codon. That's methionine. Ano naman ang ating stop codon? 3 ang ating stop codons. What are those 3 stop codons?
UAA.
UAH, UAG,
UGA, UGA.
Ayan. Thank you so much, no? At least may kausap pa rin ako, no? Okay, pero parang 3 lang yung kausap ko, 4. Okay, so UAA, stop. UAG, okay, stop din yan. UGA, stop. So, yun yung mga variety ng stop codons. So, ang tawag sa kanila ay terminator, no? Okay, so, or the stop codon, the terminator. Yung una-una, okay, we call those as the promoter or the start codon. So, yun yun. Okay, now, so yung amino acids, there are 20 amino acids. Huwag kalimutan, 20 amino acids. Okay, so each with a basic structure. Okay, so now sa amino acids na yun, 20, ang ating, screenshot nyo yung mga pangalan, mga anak ng amino acid. Okay, so ito yung mga amino acid natin. So yung ano doon, yung 11 doon, that's essential. Amino acids, yung 20 na kailangan natin. Yung 11 doon, that's non-essential. Ibig sabihin, nasa katawan na natin. Yung siyam, okay, so yung siyam na amino acid, that's essential. Ibig sabihin, kailangan mong kumain. Ang kailangan mong isupply yung protina na yun para ka lumaki, para makagawa ng panibagong protina. Kaya ka lumalaki. Okay, so subukan mong hindi ka kumain. Hindi ka lalaki at maging buto-buto ka na lang. Kasi walang protina magagawa. Okay, so yun. Okay, so once again, tuloy yung translation. Okay, so sa translation, okay, siyempre, okay, so yung mRNA, lalagyan lang ng pangalan, no? Si mRNA, kung anong na-produce na mRNA doon sa transcription, lalagyan mo lang ng pangalan. So, group of 3. So, AUG, yan yung group of 3. Start codon yan. Okay, so, then another ones, GUA. Okay, yun lang. Ganun lang kasimple yun. Okay, pero that's very complicated one sa ating, nasa ating katawan. Okay. Okay, so sa translation, mayroon 3 steps. Initiation, yung pag-umpisa. Elongation, ibig sabihin tuloy-tuloy lang. Okay, then termination, yun yung pag-stop. Okay, so yun yung start codon, then stop codon. That's for termination. Okay, so sa initiation, ito yun. It will be started with a promoter, or we call that as the start codon. Okay. Okay, so this is the initiation. Recognition site, no? Okay, so kailangan yan. Okay, then transcribe. Okay, so ito transcribe lang. So para maging ano katulad nito. AUG. Okay, so methionine yan. Okay, so then we have GG. Ano yung nakalagay? Okay, so GGA. Okay, kung ano man yun. Okay, then another one, this one. Okay, so for elongation ulit. Medyo complicated lang to, pero ano, basic lang muna tayo ngayon sa grade 10. Pagdating ng grade 11, grade 12, okay, college, medyo ano na to, medyo complicated na. Okay, so far, kayang-kaya niyo yan. Okay. So once again, ito yung amino acid na 20 ha. Yung 11, that's non-essential. Ito yun. Ibig sabihin, nasa katawan na natin to. Ito ang kailangan mong kumain, kailangan mong i-supply everyday. Okay, so phenylalanine. Could you imagine, no? Methionine, nasa ano siya, essential. So pag may pagkain ka at walang methionine, yung pagkain mo, hindi mag-start sa paggawa ng tawag dito, sa paggawa ng protina. So sa essential, meron palatandaan, private lieutenant H-link. Okay, so yun yung phenylalanine, valine, or valine, threonine. Okay, so LT is leucine, tryptophan. Okay, so then yung H, histidine. Okay, then L, lysine, isoleucine, and methionine. Okay, so yun yun. Okay, so mga dapat yung tandaan, coding strand. Okay, take screenshot mga anak. Pag sinabing coding strand, another term yan is what? Anong tawag?
Non-template strand or side strand.
Okay, yes. Pag may sa mga libro, pag may nabasa kayo, ito yung mga term na ginagamit ha. Coding strand or non-template strand or sense strand. Okay, so coding strand. Ibig sabihin, doon ka kukuha ng code. Okay, kukode mo yung mRNA. So the strand of DNA not used as a template for transcription. Okay, so this is the coding or non-template or the sense strand coding. So it corresponds to the ayun. Okay, so it corresponds dito pala yung tambay lang coding. Okay, so yung non-coding, okay, so yun yung the DNA strand that mRNA is built from. Okay, ito yung tatapatan mo ng mRNA. Coding strand or non-template strand or sense strand, it corresponds to the same sequence as the mRNA that will contain the codon sequences necessary to build proteins, runs in a 5' to 3' direction. So usually naman doon ka mag-code, okay, saan? Sa 3' to 5'. Okay. So once again, okay, yung mga term na ginagamit natin. Okay, so we have here the coding strand. Another term ng coding, we have the plus strand, sense strand, non-template strand. So, ang takbo niya is 5' to 3'. Okay, so that's the coding strand. Ibig sabihin, dito yung pinagmulan ng DNA, no, para sa replication. That's the coding. Okay, so yung product ng replication, dyan tayo gagawa para sa transcription. So ang tawag natin doon, we call it as the template strand, anti-sense strand, non-coding strand, or the minus strand. Okay, so we have here the 3' to 5'. Okay, so once again, sa transcription, okay, so ang direction na is 3' to 5'. Yung pinagmulan ng DNA replication, 5' to 3'. Okay, then gagawa na ngayon ng mRNA, the direction now would be what? 5' to 3' for the mRNA. Okay, this is the coding strand, then this is the template strand. Okay, so tandaan yung DNA, nasa loob lang yan ng nucleus, ang lalabas lang ay yung si RNA. Okay, so dito. This is the coding strand. Okay, so let's start with this one. Okay, so for DNA replication, ito yun. Okay, so the coding strand, we have ATT. Okay, then the replicated strand, ito yun, yung Na-duplicate na strand. We have what? TAC. Ito yun yun. TACT. Okay, so aga G, CATT. Yun yun. Okay, kukunin mo to. Okay, so yung na-duplicate. Okay, i-unzip mo. Okay, so once again, ito yung original DNA. I-unzip. Pag na-unzip, okay, so hahatiin mo yun. Yung na-duplicate, yung product ng replication, okay, so yung nasa baba, yun ang gagawin mo ngayon ng transcription. Okay, so magiging ano na siya, mRNA. So from T, magiging A. Okay, so papartneran mo lang. So yun na yung magiging RNA. Yan na yung lalabas. Okay, so para pumunta na sa cytoplasm. Okay, so then transcription yun mga anak. Okay, so pag na-transcribe, okay, ta-translate naman. Okay, so ta-translate na natin. So group of 3. So we have AUG, AUC, UCG, UAA. Okay, so paki-check nyo nga sa inyong genetic code. Okay, so when you transcribe, first 3 mo na first 3. AUG, AUC, okay, so yun yung lalabas. Methionine, okay, isoleucine, serine. Okay, so ang tawag na sa sequencing yan, we call it as polypeptide. Okay, so as a whole, ito na yun. Okay, so paki-take screenshot mga anak. Okay, so once again, mula sa DNA replication, okay, so i-unzip mo. Kukunin mo yung na-replicate na DNA strand. Okay, so yung na-replicate na DNA strand, kukunin mo. Okay, after unzipping. Then, lalagyan mo ngayon ng mRNA. Yes, RNA. So, paano malaman na RNA? Ang partner ni A, adenine, ay ano? Ang partner ni adenine ay natulog na, wala na akong kausap. Okay, so ang partner ni adenine ay uracila, mga anak. Okay, so transcription, translation. Okay, so once again, DNA replication. Ito yun, yung nasa taas. Sa transcription, kailangan mong i-unzip at kunin mo yung nonsense strand. Okay, so non-template strand. Okay, so para makuha ka ng mRNA or RNA. So, tatapatan mo. Pag nakuha mo na yun, baba mo. Okay, so ready for translation. Okay, so paano nag-translate? Tigi ilang bases ang kailangan natin?
Tatlo po.
Okay, yes, tatlo. Anong tawag natin doon sa tatlong bases? Tatlong nucleotides. We call those as, tawag natin doon ay codons. Okay, so we call that as codons. Okay, tato lang. AUG, methionine yan. AUC, ano ang AUC? Tingnan nyo sa genetic code ninyo, mga anak. So, we call it as AUC. Check nyo mga anak yung AUC.
Sir, paano magbasa nito?
Leucine.
Yes, isoleucine. Okay. AUC. Next, UCG. UCG, pakitingnan mo anak.
Serine.
Yes, serine. Okay. Kaya nandito ser ang nakalagay. Next, UAA. UAA.
Stop po, sir.
Stop, sir.
Okay, stop na. So, nag-stop na dyan. Okay, so, kunyari lang, na-stop na. Okay, para hindi humaba. Okay, so, that's the total description for the Central Dogma. So, ganyan lang siya kasimple, pero complicated talaga siya. Okay, so ulit, paano nangyayari ang Central Dogma? First step is, anyone who would like to explain? Carl? Carl Ivan? Okay, would you like to explain the Central Dogma? John Ray? Yes.
Wait, sir, wait.
Okay. Saan mag-umpisa ang Central Dogma? Carl? Ay, ay. Okay, so medyo technical kasi ito. Pag hindi, if you are not an open-minded, malilito ka, maguguluhan. At saka kung wala kang background ng pag-intindi sa biology, mahirapan ka talaga. Ang tendency niyan, sasabihin mo, ano ba ng biology? Ang hirap-hirap. Okay, so ako hindi kasi ako masyado sa details. Sa general details ako bumabasa. Yung kalalabasan, yun ang gusto ko aralin. Yung anong mangyayari. Una, anong step? Okay, first step sa Central Dogma is John Ray, kung hindi kaya ni Carl Ivan. Uy, wala akong kausap. Okay, so first step is what? DNA. Uy, kayo naman
Replication, sir.
Okay, yes, DNA replication. So, anong gagawin dito sa DNA replication based on this example? After ma-replicate ang DNA, anong gagawin? Anong mangyari? Pag na-replicate si DNA, kukunin natin yung
RNA.
Okay, so yung RNA, no, ko-convert natin. So, gagawin natin RNA, yung na-replicate na strand. Okay, so yung na-replicate, ito yun yung TACTA nasa baba. So, ngayon, yan yung gagawin natin ng mRNA. Okay, so AUGA, yun yung makatapat na yan. After transcription, what's next? Ta-transcribe lang to RNA. Pag na-transcribe na sa RNA, anong gagawin?
Translation.
Yes, ita-translate mo na. Paano mo gagawin yung translation? Gugrupo yung...
Ipag-prepare sila sa tatlo, sir.
Yes, okay. So, you have to extract codons, no? Okay, so group of 3 nucleotides or 3 bases, no? Okay, so ganun lang kasimple yun. Ano mo lang, group into 3 bases. Tapos, lalagyan mo na ngayon, ita-translate mo na into amino acid. So, tatlo-tatluhan lang yan. So, pag na-translate na into amino acid, yun na yung group of amino acid, ang tawag ay polypeptide na. So, yung buong polypeptide na yun, ang tawag na ay yung buong polypeptide or chain of amino acid, ang tawag ay polypeptide. Okay, now, so as a whole, in general, anong tawag sa kanila? We call it as a protein, isang protina. Naintindihan po ba? Okay, so get your notebook. Okay, so could you please give the complementary strand for this one, 5 prime. Okay, so show it on screen kung tapos na. Okay, nakopya na mga anak? Okay, start tayo ng dito. Start tayo ng Central Dogma. Okay, so nakopya na sa notebook. You're going to send it through our messenger. Okay, start tayo. Okay, so yan yung major ng DNA. So, DNA, dapat yan ay double strand. So, replicate natin. Okay na, tapos na? Ma-replicate? Hello mga anak. Nagsusulat po ba kayo?
Sir, wait lang po.
Ayan, very good. May sumasagot. Buti may sumasagot pa. Okay na?
Opo.
Okay. Patingin nga sa ano nyo, sa webcam kung tapos na. Okay, so this one. So ano magiging kapartner niya? Who would like to answer? Ano magiging complementary strand niya? Yes, John Ray. 5' to 3' yan.
TACG, TAT, CTAAT, CC, TATA, GGG, TCTATC.
Ayan. Okay, so ganyan ba? Mga anak, ang naging sagot ninyo? Okay.
Yes.
Okay, so kung ganyan, lagyan ng 20 points. Ay, kung tama. Okay, so ngayon, okay, convert the given coding sequence into an mRNA strand and a corresponding polypeptide sequence. Okay. Una ay, anong ginawa natin? Nag-replicate tayo, di ba? So ngayon, ta-transcribe nyo. Okay, so please transcribe to our mRNA, messenger RNA. Okay, so ito yung ano, ito yung sequence. Okay, please transcribe to mRNA. Nasa ano tayo, proseso ng paggawa ng Central Dogma. Okay, in your notebook pa rin. So 20 points yung una, ito 20 points din to. So wag madaya ha, pag walang gawa, wag maglalagay ng ano, ng score. Ang iba kasi nangongopya na lang. Wala naman talagang gawa. Okay, so gumawa naman please. Okay, anong gagawin lang? Kailangan gawing mRNA. Anong clue natin dito? Papartneran lang sino? Okay, papartneran lang yan si T. Anong partner ni T sa mRNA?
A, sir.
A.
Okay, yes, A. Si A, anong partner ni A sa mRNA?
U, C,
Okay.
I, U,
Yan.
V,
Okay, yes.
A.
Tuloy. Sino yung sumasagot
U, A,
kanina?
T. Clemente po, sir.
Okay. Mary Francis. Okay, go.
A, U, G, C, A, U, A, G, A, U, U, A, G, G, A, U, A, U, C, 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 A, G, A, U, A, G.
Ayan. Ang direction. Mary Francis, anong direction?
5' to 3' po.
Yes, okay. So, after this one, andyan na yung mRNA, ano nang gagawin?
Yung sa codon po, sir.
Yes, codon. Okay. So, para sa paggawa ng codon, anong gagawin? I-group sila by?
3, sir.
Okay, yes. Group by 3. Okay, yan na. Okay, go. Okay, so group of 3 na yan. Okay, hanapin ang tawag dito. Pakihanap ang genetic code. Okay, lagyan ng okay, so anong dapat ilagay? Okay, mga pangalan. Okay, so lagyan na ng mga pangalan ng amino acid. Okay, paggawa nyo, ipopost nyo maya-maya ha. Okay, so dapat ay post agad, hindi yung kinukopya lang yung sagot. Gumagawa ba kayo mga anak?
Yes, sir.
Yes.
Ay, nako. Please do. Try lang, haba try. Kung medyo mahirap, intindihin niyo talaga. Kailangan niyo talaga magbasa. Okay. Pero anyway, sinasabi naman. Okay. Genetic code lang naman ang kailangan mo dyan. Okay. So, pakilagay sa chat box yung tapos na. Just type done. If you are done. Okay. Tapos na po ba? Okay. Tapos na? Okay. So, anong mga amino acid ang nakita nyo? Okay. So, that sequence is called...
Tapos na, sir.
Okay. So, yes, Ron. Okay. Sige nga, Ron. Anong amino acid, Ron? Anong nakita mo? Okay. So, yung tapos na, pakipost na at sa ating GC. Okay, so that would be the end of our lesson today. Okay, so I hope natutunan. May natutunan kayo sa Central Dogma. So yung mga lesson na hindi ko na discuss, I will just be posting a video or YouTube video for you to watch. Okay, so question? May tanong? May tanong mga anak? Uy, may tanong? Okay, assignment. Ito yung assignment ninyo. Okay, ilalagay niyo sa e-portfolio ha. Submission ng e-portfolio Friday. Okay, so assignment,
Sir, nakamute ka po ata.
Ayun nga, sorry. Salamat ha. Thank you. Okay, so assignment. Okay, nakikita niyo assignment? Paki-screenshot yung assignment niyo. May nakikita kayo?
Meron po, sir.
Yes po.
Okay. Paki-screenshot. Okay, so yun po yung assignment niyo. Na ano na yan ha, kunyari ano na tayo, tapos na yung replication. Ano na tayo, tawag dito, ano na, tapos kunyari tapos na yung replication, proceed na kayo sa transcription at saka translation. Okay. So that would be your assignment. Okay na, na-screenshot na po?
Yes po.
Okay, picture taking. Okay, please open your webcam. Bagal ng internet ko. May naka-open na ba ng webcam? Ayun.
Yes, sir.
Okay. Nakikita ko dito si Mary Francesca, John Ray, Jasmine. Okay. Nazizikay na. Buhok ang nakikita ko. Uy, si Kaina, ayusin mo yung camera mo. Buhok mo yung nakikita ko dito. Hindi yung mukha mo. Ayan. Okay. So, ready. 1, 2, 3. Ayan. Thank you so much, mga anak. Okay, ingat palagi. Okay, that's the end of our third quarter. Okay, so next week, fourth quarter na tayo. So good luck. Sa integrative, di pa ako nag-grade ha. Okay, so baka this week. Mag-grade ako. So bye-bye. Bye-bye sa inyo.
Bye, sir. Thank you po.
Bye, sir. Thank you po.
Okay, ingat palagi. Ay,